Good afternoon, and welcome to today's BNA Credibility Lunchbox series in vivo Neuroscience. My name is Emily Sena, and I am your host for this series. This is the latest in our ongoing in vivo webinar series that the BNA is running as part of our work to strengthen the credibility of neuroscience. And we're very grateful to the Gatsby Foundation for their support. We held 3 webinars back in March, and this is, I guess, the second season. We had one last week, and then today we have a speaker who's going to speak about data handling. These are very short webinars, they're only half an hour. The format is that our speaker will speak for 10 to 15 minutes, and then we'll have the rest of the time for questions. I'll be very grateful if you have questions, and you can type them as the talk is going on to enter them into the Q&A box, and you can use the chat function for just general chat. But if you want questions post the speaker, then please use the Q&A box. I should also say that this webinar will be recorded, and along with the other webinars in the series will be available on the BNA's Credibility in Neuroscience website. So it's my great honour and pleasure to introduce this week's speaker, Dr. Kaitlyn Hair. Kaitlyn is a preclinical meta-research scientist with a background in psychology and neuroscience. I should disclose that Kaitlyn and I are colleagues here in Edinburgh, both members of the CAMARADES group. I should also mention that Kaitlyn was one of the speakers when the CAMARADES group won the Team Credibility Prize last year. And she's been, I guess, the tour de force in our group in making sure that we keep straight around data handling. Kaitlyn is also a member of the university's reproducibility working group, so working to encourage reproducible and credible research across different disciplines in the institution. I will stop speaking and hand over to Kaitlyn, who is going to present about data handling. Thank you, Kaitlyn.
Thanks, Emily. That's great. It's really nice to be here today. Let me just share my screen and hopefully we can get the slides. Here we go. Great. So as Emily said, today I'm going to talk about handling data in your in vivo studies. Let me just minimize that slightly. So why does data management actually matter? So first of all, it's about transparency. So if you have a data management structure throughout your project and you plan for it, it makes it so much easier for you in the future to go back and understand what you actually did, what your processes were, how your raw data got transformed into your final results. And it's also useful for any collaborators or future colleagues, anything like that. You can go back and actually understand, hopefully, what your data folder is and what's going on. And I love this quote at the bottom of the slide here, that your primary collaborator, let me say that again, your primary collaborator is yourself 6 months from now, and your past self doesn't answer emails. I just love this quote from Twitter because I think it's so true, and I think we've all been in that position where you step away from a project for, it could just be a couple of weeks or it could be 6 months. You go back and you just have no idea what you were doing and what all the files mean. I think having a good data management plan also enables and encourages sharing your data, code, methodology later on. You'll have more confidence in how you actually got from A to B. And in that way, I think it's a really vital part of the movement towards increasing rigor and reproducibility in our research. So we all probably know this feeling when maybe you've collected your final data point. So let's say in this example, you're doing a behavioral experiment in animals, and maybe you've just measured the last animal and now you're ready to analyze your data, which is quite an exciting point in the project. Maybe you didn't collect the data, maybe it was one of your students or a collaborator, and you're looking at the folder and it looks something like this, where you've got all these data files, final, different versions. And your first step is you need to actually find the right data set to analyze and bring into whatever analysis software you're going to use, whether that's R or something else. Then you try and do your analysis, and maybe you end up with lots of files again, because we all know how frustrating it can be to get your script to run and your analysis to work. Maybe you generate lots of different plots with different axes, and maybe one is in certain colors. So very, very quickly, without a plan, your folder can look like this for the analysis. And then when you come to write the paper, you need to make sure you're selecting the right plot and the right final results that you want to include in your paper. Maybe then you need to go back to that original folder and find your protocol somewhere, the correct protocol, and put that into your methodology. And all of this can just become a bit of a headache, and it's not a nice feeling when you get to the end of the project and have to piece it all together like this. It doesn't make me feel very confident when I'm in this position. So ideally, you really want to somehow go back in time and start with a plan. And if you're about to start any new projects, this is definitely for you. So I think the first step is to create a project folder, which might seem simple enough, but also create the subfolders before you start. And I think this has a really powerful effect, because if you have the subfolders there, so this is an example structure on the right-hand side with things like methodology, raw data, results, analysis, manuscript. When you're going through the project, if you have those folders, psychologically, it makes you more likely, I think, to put them into the folders. So having that structure right from the start is really helpful. Don't just keep everything in one folder is a top tip, because that's how you end up with just really messy data and everything everywhere. Another top tip is to retain all of your raw data. So if you think about your raw data, it's kind of, you want to have it for later on. If you ever need to go back and see, maybe you made a mistake in the data processing or in the analysis, you really want to have that traceability back to that original raw data file to be reproducible and to be certain about your research. And ideally, if you can, you should make those raw data files read only, so that no one can accidentally go in and modify something. Another top tip is to have what's called a README file, which you may have heard of. This can be anything. It can be like a Word document or a text file, whatever's easier for you. But it basically documents in a lot of detail what's in that folder. How the files are named, what it actually means, what's in your raw data folder. This can be really useful. I'll give an example. So imagine you go into the lab one day and your calibration isn't set up right for some behavioral experiment, and you're getting strange results, and you maybe have to measure the same animal 3 times. In 6 months' time, maybe you write a little note to yourself somewhere that this happened, and that's why you've got 3 versions of the same animal. But in 6 months' time, if you go back to that data folder, you probably aren't going to remember why you have data like that and what it all means. So it's that level of detail that you put in the README file, and it needs to be continuously updated going through the project, where you make these little notes to yourself for the future, and also, of course, for any collaborators that want to come in and pick up this file and use it. So it's in quite a lot of detail. And I'll talk about electronic lab notebooks in a minute, but this could also be replaced with an electronic lab notebook or any other sort of documentation process that you can use throughout the project. And this for me has been like a really big change in the way I do things, and this is something I've adopted in the past year or so, and it's made a really big difference to being able to go back to projects and actually understand what I was doing. How you name your files obviously really matters as well, and this is quite a specific example where they have a lot of information in this file name, and your files might not need to be as specific as this, so it's worth thinking about what sort of data you're going to generate and how detailed the files have to be. The convention is to start off with general information about the file on the left-hand side, so information like the date, moving to more specific information on the right-hand side, so information about maybe the experimenter that collected the data, or the animal ID, or the version number of that data. And that's just sort of a convention to work with. I think starting with the date in general is quite a good idea, because it means, especially if you put the date in this format, so you have the year, so 2013, the month and the day. If you do it in this format, it means you can sort your files by date in the folder, which is just really helpful for going back and looking at the data. it might go without saying, but you also really need to keep your data safe. I think everyone's heard of these disaster stories where someone loses all their data, and it really does happen. And when I was doing my PhD, I got given this really old computer that was sourced from another lab. And it was really funny because I always remember the technician that set it up for me. He just looked me in the eyes and was really, really serious and just kept saying, "This computer can die at any time. Anytime. Don't put any data on this computer. It can die at any time." So I don't know if that computer was just particularly, on its last legs, or whether he's seen this happen before, but it really does happen, and any computer can die at any time. So please back it up on some sort of server, cloud-based service. I think it's recommended that you back it up in 3 places, but I know that's probably overkill, but yeah, definitely back it up somewhere. I thought I'd just briefly mention electronic lab notebooks. So if you're currently keeping track of your project with some sort of written notebook and your own handwritten notes, I think it's worth thinking about the benefits that electronic lab notebooks could bring. So first of all, they're searchable, and usually they have online and offline searching capabilities as well. So you can search through your notes for certain words and phrases rather than relying on flipping back through a notebook. You can usually export your data as well in different formats, which can be quite useful. You can share your data with collaborators and the broader research community. Obviously, it depends how shareable your data is, and the different notebooks maybe have different ways to do this, but most of them do allow for collaboration. You can also embed really high resolution images, different types of data. There's different notebooks specialized for different types of data as well. So it's worth looking into this and thinking about what one might be most useful for you. You can usually access them globally as well. So if you're going to visit a collaborator overseas, doesn't really make sense to bring your handwritten notebook. You can see why it might be useful to have access to some sort of online notebook that you can actually share with those collaborators. Also, a lot of these notebooks now have mobile apps. So if you're not able to bring your laptop or computer into the lab, and maybe you are still writing some handwritten notes, you could take photos of it on your phone and upload it to the notebook as well. So there's lots of integration with things like that. With all of these strategies, so with the notebooks and the documentation and files and folders, ideally you should have a lab strategy and not just an individual one. I think this is more useful because it means everyone adopts the same approaches. It's a bit less daunting than having to try and think of all this by yourself. It's better for continuity because if everyone is following sort of similar structures and similar processes, it means that if you have students coming into the lab, they should adopt that too. And it means that their project should be easily understood later on if someone wants to pick it back up. And also if there's staff turnover, ideally, it means that you should be able to go back to that. If you get a new postdoc, they should be able to understand the previous postdoc's work and carry it on. And I think by everyone adopting these approaches, it also keeps everyone motivated, because something I haven't really talked about that much is that all this stuff does take time. It takes more planning, more time investment. I think you gain a lot at the end, but it does take time. And I think everyone working together on it keeps everyone motivated. I just wanted to briefly talk about data workflows as well, and about how having your data in this sort of structure can help you share your data and workflow later. I know that next week we're going to have a BNA credibility seminar on data sharing, so I'm not going to talk about it in detail, but basically if you have a folder structure like this and you've managed your data well throughout your project and you've managed all your files, ideally, you should be able to just upload that to some repository like the Open Science Framework, for example. And if you have analysis scripts in there, ideally, they will link back to that folder structure because all of your file paths will actually match up. Of course, this depends on how shareable your data is and things like that, but this is in an ideal world. It does, I think, make it a bit easier to share at the end. And I think another benefit is that, for me anyway, having this sort of open workflow and sort of working towards knowing how your data got from the raw data, how it was processed, how it turned into results, it really helps me feel confident in my work and that I've done things properly. And that also makes me more confident to share the data. Moving towards the future, I think these sort of things are worth considering, because there's now something called executable research articles, which some of you might have heard of. And basically what they are is dynamic documents that contain text, data, and analysis code. So it's kind of a publication that contains all of that within it. So they're fully reproducible is the idea, a lot more so than typical journal publications. And if you use something like R, you can use a package called R Markdown to basically turn your R scripts into these nicely formatted publications with text and code chunks that also print out your plots as well. So you can understand, how was that plot generated? What's the data behind it? How does it all link together in a reproducible workflow? And there's a similar thing for Python called Jupyter Notebooks that you can use. And these are now being accepted by different journals. I think the one that I know more about is eLife. So they've got these executable research articles now, and it's great because you can go onto these publications and they have a little button beside the figure, and you can click down to actually see the script behind how that plot was generated. And I think, in the future, this could become more common. So as a summary, data management definitely shouldn't be overlooked. I think it sometimes is in terms of reproducibility. It's an integral part of doing open and reproducible research. It does take some more initial planning and time investment, but I think it is worth it in the time you save later. You don't have to search as much. You don't have to do as much, opening Excel files and trying to work out what you did. And I think you have more confidence in your own data and workflow as well. I think collaborative group approaches definitely work better than individual ones. So it's worth, if you're a professor or sort of higher up in your lab, you should think about what you can do at a lab level to implement this, even if you're not doing experiments yourself anymore. And there's lots of tools out there that I don't have time to go into, lots of electronic lab notebooks and other tools that I've mentioned. So it's worth looking into the right one for your type of data. Thank you. So I'll stop there. Stop sharing my screen.
Wonderful. Thank you. As usual, very informative. I'm always learning. So we'll kick off the Q&A. I just want to remind people, if you do have questions for Kaitlyn, please put your questions in the Q&A. I will take chair's prerogative and ask the first question, and then go through the others. So, as I mentioned, obviously we've worked together for some years now, and you will remember that we have in the past spent time stalking ex-students and ex-lab members on Facebook and trying to get their contact details to ask them what they meant by this and all that kind of stuff. Despite some people in the group saying, we need to have better mechanisms. And I guess the issue is a cultural issue in a lab. Like, I think the arguments are convincing. To me, at least to me anyway, it's a bit of a no-brainer. Yeah. But you're trying to change people's behaviours and change cultures in the lab. And I just wondered if you had any thoughts or tips that people could employ or need to think about when trying to implement something like this in their research groups.
Yeah, so I think a lot of it is about changing the mindset, because I think right now, I think a lot of us work in these sometimes quite small teams, and people come in and it's sort of like, maybe you show them on a computer. When it wasn't COVID, you could actually show them, this is how we do things, and it was very closed off. Like, it was just, this is how we do things in this small research group, and we produce these research outputs, papers, and that's what we do. And I think it's about thinking from the start of the project that you're not just a small research group, you're going to share your research with the world potentially. So I think it's kind of shifting it to think, how would someone from a completely different lab do this? It's kind of opening up the whole workflow from the start. So I think, it's a difficult
question. I think if we knew how to change cultures, then I think we would. Yeah.
We might not be here if we had an answer, I guess. But I think having everything, online as well, like, we don't need to, I think we have a tendency to just have these little guides on how to do things, maybe on our computer, or, speak in our lab as well, like speak to this person because they know how to do this, but we don't actually, we don't put all that somewhere online, like a sort of, this is how we do things. Yeah, resource. Yeah, I think for anyone who's wanting to implement this in their lab, it's about trying to create a resource like that and pulling together all the sort of knowledge that's already there on how we do things into some sort of resource. Yeah.
Thank you. So we have a question saying, would you please recommend some electronic lab note applications websites? Are they free?
So some of them are free. I think honestly there's too many, and because I don't actually work in an in vivo setting, it's kind of hard for me to recommend what would be better for different people, because I think it really depends on the sort of data you're working with. I think, I mean, in our lab, what we've sort of done, because we're just working with, I guess, like we do sort of systematic reviews and that sort of thing. So our data is very different, but I think we just use sort of OneNote and these sort of tools that are actually free and exist. But I think, to be honest, even having the README file as a start, just within each folder, and some sort of documentation, no matter what it looks like, is better than nothing. Especially if you're sort of working from paper just now. But I will post in the chat, I did find this really good sort of analysis of all different lab notebooks, because someone has done like a really detailed analysis of this actually for different people. So I'd recommend having a look at that and working out what's best for you, depending on your needs.
And I think it's also worth checking what your institution has bought into. So you said OneNote's free, but it's actually only because the University of Edinburgh has this subscription.
Yeah.
So to us, it's not additional. And I think at Edinburgh we also have, I think we have a subscription to RSpace as well. So which is more directed to wet labs. But it's worth going to your information services, digital sort of resources people in your institutions, and seeing what have they bought into. I think often institutions do have subscriptions to stuff, but people on the ground aren't aware of them and aren't using them. We have a question from Elena Christina. Great talk, Kaitlyn. Similar to Emily's question, I was wondering if you have any thoughts on how to handle a situation when two collaborators have very different systems on organizing their data, or even inside a lab if each person is using a different system.
Yeah, I mean, I think we've had this issue. Yeah, we've had this issue, and I guess we're like still trying to change it just now. I think it's very hard when if you're already partway through a project, it's quite hard to sort of go back and change it, I think. But I think at least during the project is the opportunity to try and do something. So even if you try and just agree on even like what the workflow is, like even if you have basic folders, right? Like just the most basic thing of like the raw data, is it somewhere, is the process data somewhere, is the analysis code somewhere? I think it's just trying to think of the easiest thing to do while you're actually doing the project, and avoiding kind of having the project finished and then having to try and go back to it later and work out what to do. But I mean, it does just literally take time. It takes literally sitting down with the person, probably working out what data they have, how you can maybe put it in some shared folder together. But it's definitely not easy, and we have this just now for a lot of historical projects, that it's going to be very painful to try and go back and understand now.
I agree, and I do think that there's something in the project leader. If you're the PI, I think you do need to set the tone. I've become much more, I guess, dogmatic and direct about this is how we're going to do this. And you get some people, even things like using OneNote or the online version of Word, and they're like, oh, I can't. And I was like, okay, let's work through it. So you do they are some people who want to do what they've always done. And you do have to kind of take a one-to-one approach with those people to bring them on side. But people using different systems in one project is just a recipe for disaster. as we have learned the hard way.
Yeah, and I think if you also say, I mean, I don't know if you're planning to share your methodology or your data later on, but I think it's worth saying that sooner rather than later, because I think if people know, and I think this is what I was trying to say, like if people know the data has to be shared later, you know, you need to understand it yourself. It does need to be in some sort of format. You can't really just put something up and that no one can understand. So maybe that's an incentive to work out what to do.
We have another question from Nina. Thanks so much, Kaitlyn, super useful. I think the culture change is spot on. Over the years, I've seen this really depends on leadership and example set. Setting up a new lab is a great opportunity to drive that change from the top. Is there a one-stop shop online for new group leaders wanting to implement a lab-wide data management strategy? That is a very good question.
So I've actually been to a talk that was on this, and I can try and I would need to go back and try to find this talk and this person, but basically it was at an Our Ladies seminar, and it was like a new PI, and she had come in and literally done this. And I'm trying to think what her tips were. Even I think she'd used a lot of R, so she created an R book down, which is what we're trying to do just now for our lab, where it's basically like a sort of document written in R script that's it turns it into a book, a bit like R Markdown, and you can update it, you know, constantly and add things to it, add complex plots and stuff if you do a lot of really, you know, complex analysis that you need to explain to someone. And that's how she did it. She basically set this up, and it meant, you know, when students came in, they had to read this, they had to follow it, they had to, you know, adopt all of the processes right from the start. Yeah, so I think that was how she did it basically. That was yeah.
Yeah, I think what you said about it being a living document, that's really important, because these things change over time. And I think actually having these what we've called the CAMARADES cookbook is, it's not just, I mean, the data handling is also what we're talking about here, but I think, you know, it's really about what the culture is in your lab, and how, you know, how we speak to each other and how we treat each other. And I think it can be a much broader, all-inclusive document about the workings of that lab. But yeah, if you can find that link, I think that would be very useful to lots of people. Fab. Okay, well, I think we are reaching time. So I just want to say a big thank you, Kaitlyn. A few people have already said how useful that was, and as somebody who's been in this space and doing this for a little while, like I say, I'm still always learning and finding this stuff really useful. So thank you. I just want to, so I'm just checking my notes here. First of all, thank everyone for joining us. It's great when we've got audiences who engage and for asking questions, because I think, you know, questions that you're thinking of other people have. So thank you very much, everyone who's attended and asked a question. For those of you interested and weren't able to attend our previous webinars, or interested more generally in the credibility work that the BNA are doing, then please check out the Credibility in Neuroscience site on the BNA's website. We will have the last or the third in this current series of the in vivo credibility webinars next Thursday at 1 p.m. Our speaker will be Matt Grubb, and he'll be taking this data piece further and talking about how we share and disseminate data that we have collected in our studies. So with that, I'll thank you again, Kaitlyn. Thank everyone for coming, and thank the Gatsby Foundation for their ongoing support for this work.
Thanks, everyone.
Cheers.